Thanks for watching this final video for our hemoglobin drug synthesis and assessment experiment. Today we're going to be looking at the analysis portion of the experiment. Of course, don't forget that your goal is to identify those drugs that bind to the active site of hemoglobin, and through doing this, I hope you'll gain exposure to medicinal chemistry, get to practice some drug synthesis, execute a very cool protein assay, and today we will get to analyze our data. Also, of course, recall that there are three main parts of the experiment, and today we're looking at the analysis portion. So last time we took a detailed look into how you are assessing how well your drug binds to the apohemoglobin protein, which is just hemoglobin with an empty active site. Recall this is done by taking the fluorescent ANX apohemoglobin complex and displacing or kicking out that ANS compound with your drug. This results in a loss of fluorescence, so hopefully you got a nice dose-response curve that begins with a strong fluorescent signal from the AHP ANS complex, but when that ANS gets kicked out by our drug, then we should have seen a loss of fluorescence. And today we'll discuss just how we will identify that IC50 value, or the concentration at which we saw 50% inhibition, or in our case binding. And this is important because the IC50 value is the measure that we use to compare drugs to one another. And the lower the IC50, the more effective or the more potent the drug, because we need less of it to get the desired effect. So in order to thoroughly analyze the different drugs, we will be discussing today what causes those IC50 values to be different, as in why a drug might be a better binder than others. And then I'm going to walk you through just how you obtain your dose-response curve. Alright, so what causes those differences in the IC50 values? Why might a drug have a lower IC50 value than a different one? It's all about those binding interactions between your drug or your molecule of interest and the target protein. When you have a greater number of and stronger interactions between the drug and the target protein, this results in a better binder and a lower IC50 because it will more efficiently kick out that ANS molecule. And of course, conversely, if you have fewer and weaker interactions between the drug and the protein, this results in a weaker binder or a higher IC50 because it will take more of the drug to kick out the ANS molecule. Alright, so what kind of binding interactions are we talking about? We're talking about reversible, non-covalent binding interactions. And we've seen these before, even way back in Gen Chem. Okay? Good examples are hydrogen bonding interactions, ionic interactions, van der Waals or hydrophobic interactions, and finally pi effects. So we're going to talk about all of these today. First up, one of the strongest binding interactions is that of hydrogen bonding. This is when you have a hydrogen bound to an oxygen or a nitrogen. So here's an example. And this hydrogen, because it is bound to a more electronegative atom, the hydrogen is partially positive, and so it is attracted to more partially negative atoms, or more specifically atoms with lone pairs. So we see this all over the place in biology, including when your DNA base pairs match up. Okay, we can see this hydrogen bonding taking place. And here's another example between two different amino acids, arginine and aspartic acid. You can see how the hydrogen bound to a nitrogen atom is coordinated or attracted to the lone pairs of another oxygen atom. Okay? And these interactions are definitely taking place between the native heme binding molecule and the hemoglobin active site. See here these carboxylic acids, here they are in stick form, and these carboxylate groups are definitely participating in hydrogen bonding between different amino acids of that target binding site. So another strong binding interaction that can take place between a drug and its target protein is that of an ionic interaction. This is just an electrostatic attraction between oppositely charged ions or groups. We have a perfect example here of a positively charged quaternary amine being attracted to the negatively charged portion of a carboxylate ion. And this is another type of binding interaction that is definitely taking place between our native heme molecule and the active site of hemoglobin. Okay, it's the positively charged iron 2+ ion is attracted to the negatively charged deprotonated nitrogen atom of this histidine amino acid of the active site. You can see how they are attracted to one another. This results in a pretty strong non-covalent binding interaction, an ionic interaction between the two. Weaker but still notable non-covalent interactions are van der Waals or hydrophobic interactions. And these are just the weak attraction between nonpolar molecules or groups, and they result when we have a polar solvent such as water. We know that like dissolves like, and therefore the water does not dissolve the non-polar regions all that well. So resultingly, those non-polar portions of a molecule will kind of stick together because they're not dissolved by the water very well. And so this results in a weak attraction of those non-polar regions. And these attractions are again definitely taking place between our active site of the hemoglobin protein and the native heme molecule. See this valine amino acid of that active site? This is a relatively non-polar amino acid because it's got this isopropyl group hanging off. It's definitely attracted to the more non-polar or hydrophobic region of the porphyrin rings of our heme molecule. And lastly, pi effects are extremely prevalent among drugs and their target proteins. These are just the attraction between the pi clouds of different aromatic systems. You can see there are different modes of binding, including the displaced, edge-to-face, and the sandwich modes. The sandwich one is the least prevalent because the pi clouds actually repel one another. So it's the displaced and the edge-to-face that are much more abundant. Here's a really good example of the phenyl ring of one amino acid being attracted to the benzene ring of a tryptophan amino acid. You can see how those rings are displaced. And lastly, this is definitely taking place in our hemoglobin active site, okay, and our native heme molecule. You can see how the benzene ring of this phenylalanine amino acid of the active site is attracted to the aromatic portions of the porphyrin ring of the heme molecule. So you can see then how all of these non-covalent binding interactions are contributing to the awesome binding properties of the heme molecule because it has so many of these binding interactions. One last topic of drug protein binding that I wanted to cover is that of allosteric binding. And this will definitely affect the IC50 because it's binding that takes place at a location that is not the active site but still results in the desired effect. So what we want is our drug to directly displace ANS from that active site of the apohemoglobin protein. Okay, we want it to kick it out directly and result in the loss of fluorescence. But it's entirely possible that the drug binds somewhere else on the protein. So here we can see it's like an allosteric site is what they call it. Here it's not binding to the active site, but it's binding to a different site of the protein. But this still results in a conformational change of the protein which could kick out ANS. And it's very difficult to tell whether or not this is taking place in our assay, right? We can't go in and directly see how the drug is binding. But when we do kick out ANS, it is a good indicator that we are binding to that active site, but we can't rule out the possibility of this allosteric binding. Okay, so in summary, again, what causes those differences in the IC50 values? It's the number and strength of the binding interactions between the drug molecule or the molecule of interest and our target protein. Again, the greater the number and the stronger the interactions, the lower the IC50 value, because the more efficiently it will kick out our ANS molecule. Meanwhile, the fewer and the weaker those interactions will result in a larger IC50 value. And we also can't rule out the possibility for allosteric binding, which causes the ANS molecule to get kicked out. It's really hard to discern whether or not this is taking place, and it's definitely a possibility. So now I'd like to walk you through just how we are going to determine your IC50 value for your drug. If you open up your HB Drugs Excel sheet, you'll notice that we have different tabs along the bottom. And the solution prep tab is what we used to make your solutions. But if you click on the fluorescence data tab, you'll open up this sheet. So we're clearly going to focus on this empty column here. Just input those fluorescent signals that you wrote down in your notebook. Okay? Input the signal corresponding to its correct sample. And after you input your data, you should notice that two other portions of the sheet automatically populated. Okay? This column over here automatically populated, and then your graph should have automatically been drawn as well. So if you take a close look at your graph, you'll notice that we have the fluorescence signal on the y-axis and then the log of your drug concentration in micromolar along the x-axis. So we use the log of the concentration because it spaces the points out nicer. If we didn't use the log, then the points would be grouped up into portions of the graph and it wouldn't look like a nice dose-response curve. So it's important that we always take the log of your drug concentration. But from here, you just need to go online to this IC50 Calculator website. Okay, this website's provided on Blackboard, and when you go there, it should look like this. And so what you're going to do is you're going to take your data from your HB Drugs Excel sheet, the highlighted portions in peach or orange. Just copy and paste both columns of data right from your Excel sheet into the website here in this Data 1 window. Okay? After you do that, the data will automatically get processed and it'll look like this. And then importantly, you want to click on that Calculate IC50, that orange button, golden orange button. After you do that, you're going to get a really nice dose response curve. I love this website. Hopefully your data looks nice and it fits to this nice curve, and then the website does all the work for you in calculating that IC50. So then what you want to do is take a screenshot of your curve and your IC50 value, because then this is the concentration at which you obtained 50% inhibition or binding with your drug. Okay, so the lower the IC50, the more potent your drug, because the less of it we need in order to get the same effect. Okay, so this number is very important. So as I said, you want to take a screenshot of your curve as well as this IC50 value. Okay, just put it in a word processing document, and this is going to comprise your preliminary lab report. Okay, so this is where we're going to compile all the data. So in this preliminary lab report, you have to include your name, the structure of your drug, and then that screenshot of your curve with the IC50 value. You just go submit this onto Blackboard, and then once all of the results are in, your instructor will compile all of your IC50 values and send out that data to everybody once it's ready. And after that, you will need to complete your final lab report. And so these will differ based on your instructor, so see the details provided by your lab instructor. Thanks for joining me. I hope you enjoy this experiment. Feel free to send along any questions. Thanks.